All right. Back to that. All right. I have to redo the arrow. So today we're going to start talking on really the last major group, well, not quite the last major group, one of the major groups of structural features or structural objects in living cells, and that is membranes. So we've talked about nucleic acids and how wonderful they are for information, storing information, transferring information. We've talked about proteins and how wonderful they are for doing lots of little functiony type things, very diverse functions and so on. All of that would be useless if there was no way to keep it all together. And so membranes of some kind or another are absolutely essential to have anything you think of as a living cell. And they're logically necessary for a living anything. So what are some of the things that membranes actually do? Well, they use this barrier function first and foremost. So there's an inside and an outside to the cell. No boundaries, you're just part of the big booming, buzzing confusion. Membranes are involved in electrical transmission. We're going to look at that specifically in nerves for a little bit, but in fact, almost all cells are electrical. The behavior of nerves is just a specialized case of the kind of electrical behavior that most cells can undergo. Since we have a barrier around things now, there has to be some way to transport. There has to be some way to get good stuff in and bad stuff out. Most cells are not completely antisocial. Most cells have at least some moment in their life when they want to communicate with other cells of the same kind or of different kinds. And so a lot of the signaling that goes on is mediated through the membrane. It's like the guard post on a country's border. You let somebody come through and talk. And there's recognition. In other words, if you're going to make a whole body, do the parts of the body know how to hold together? A lot of that recognition goes on at the level of the membrane. So when we think about this, we see that the structures that are needed to perform those functions would ideally have the following properties. They'd be self-assembling, so you'd always have something around a cell. They would be self-healing. If membranes didn't heal essentially immediately, cells would leak out their guts whenever you twitched. And they need to be semi-permeable. That is, they need to let some stuff in and out and not other stuff. And a particular example here is one that's focusing on two very important components that membranes mediate the movement of sodium ions and potassium ions. And so if you look at a typical cell, there'll be more potassium inside the cell than outside. There'll be more sodium outside the cell than inside. And since the natural tendency would be for the sodium to come in and the potassium to go out, that membrane is responsible for seeing to it that things don't just average out into a mush. We're going to see how it accomplishes that over the next few days. The essence of performing what lipid bilayers do, membranes do, depends upon the existence of a lipid bilayer. And it's called a lipid bilayer because there's two layers of lipids that are involved. And they can line up so that their hydrophobic bits are in the middle of that bilayer, and their polar charged outer bits will be in contact with the water. So you can see here you've got a charged group on the head of this palmitate, and then this long hydrophobic tail. And so when it's organized as a lipid bilayer, the hydrophobic side chains can be tucked away from water, and the charged or polar head groups interface quite happily with water. They are said to be amphipathic. They feel both ways about it. So can you think of an amphipathic amino acid? You can think about it tonight. Because of the chemical properties of this, it will form spontaneously. It does not need any energy input. It does not need any template. It simply happens because of the properties of the molecules themselves. And similarly, because this is a consequence of just the hydrophobic effect and the packing away of those side chains from water, it will be self-healing. If you pierce it with a needle or something, it'll flow right back in and close back up. So this is a very handy structure to begin with as kind of the scaffolding or the basis for membranes in general. You've seen this schematic many times or something like it many times, and it's a very handy way to represent it. It does get across the idea of the fatty acid side chains and the head groups. It is a simplification, and I want to just point this out to you because we use these simplifications in a number of contexts, and it might be especially a simplification in the case of the membrane. So this is a more realistic representation of a membrane that's been generated by molecular modeling. And you can see how here in the interior, you've got the carbons of the palmitic acids tucked away from the water. The reds here are oxygens that are part of the head groups. The browns are part of nitrogens that are part of the head groups. The green is actually water. The orangey stuff here is the phosphorus. And one of the things that should come out of this for you is the tangled character, the irregular character in the interior. So if you look at these fatty acid side chains, they're not all nice and tidy like this. They're kind of flopping around like a bunch of animated spaghetti. And that dynamic character is important to keep in mind when you think about what membranes can do and what they can't do. It's also not perfectly flat. I mean, it's on average flat, but it's not going to be perfectly flat. Any questions about the simulation before we go on? Okay. Although we talked about that as if it was made out of fatty acids, we don't see free fatty acids very often in biology. They work as detergents for the most part, so you're not going to want to have free fatty acids very often. Some of the fatty acids have double bonds in them, and so they are unsaturated fatty acids. This is the fat part. That makes it an acid part. That's the unsaturated part. This particular one is called oleate because it comes from olive trees. And this is linoleate because it comes from flaxseed. It has two double bonds in it. And since these things by themselves would be pretty good detergents and break up a cell, there are not very many of them available like that free. Instead, they are often in chemical compound with a carbon backbone. And the most common carbon backbone that you're going to find fatty acids complexed with is the glycerol backbone. So here is the glycerol molecule. It's a 3-carbon molecule, three alcohol groups hanging off of it. It's called glycerol because it's an alcohol, OL, and because it tastes sweet. Many of the candies you eat probably have some glycerol in them. And if you want to keep your brownies tender, I suggest adding some glycerol. It's good stuff. Perfectly innocent. And so what you can do is you can attach the fatty acids to the glycerol backbone through the acid group, making an ester here. And now you have fatty acyl groups. It's not a fatty acid anymore because it's in a compound, so it's a fatty acyl group, carbonyl, oxygen there. And this particular one is a triacyl glycerol, so each of the carbons has been occupied, each of the alcohols has been occupied. And this compound, as you can imagine, would be a pretty neutral compound, and it turns out to be a good way to store fat when you don't want to use it at the given moment. So this is the most common way in which you're going to find fat in a fat cell. Another big group of lipids are the glycerophospholipids, and the name is telling you what we're going about. Glycero, so it has a glycerin or glycerol backbone. It's a phospholipid, so there's a phosphate in there somewhere. And then lipids, so we can expect there to be nonpolar groups hanging off there somewhere. And what it looks like is this. It looks like a glycerol backbone, the 3-carbon glycerol backbone. 2 of the 3 alcohol groups are occupied by fatty acyl groups. And the third one then has a phosphate group with something. And what that something is will affect the chemical properties of it, will affect how useful it is in membranes and so forth. So here is a phosphatidylcholine, because the something on here is the choline molecule. And here's a phosphatidyl ethanolamine, because this is ethanolamine. These two by themselves would be ethanol, but there's the amine group, so it's ethanolamine. Here's phosphatidylglycerol, with the polar group hanging off as glycerol. Phosphatidylserine. So you can see here, I think, that this is the amino acid serine that's been slapped onto the phosphate group of the phosphatidyl lipid. Now, if you take a look and think about it, not only are there different groups in here, but they are different groups with a range of properties. So here's a bulky positive charge. Here is a positive charge, but not so bulky. Here is a neutral but polar group. And here is one that has both a positive and a negative charge on it. So you can tweak the properties of these glycerophospholipids by changing the properties of the head group. That'll affect how the head group interacts with the rest of that polar part of the lipid bilayer. You can also affect it by what you have as the side chains, what fatty acids were used to make the fatty acyl side chains. Like everything else in biology, the shape of the molecule will determine the final structure. And we see this very much in the case of lipids. So if you look at the shape of, for instance, a fatty acid, its head group is larger in terms of the minimal space it can take up in its tail. And so fatty acids by themselves don't pack very well. The tails there aren't enough tails for the space taken up by the head group, so they don't pack very well. One with two fatty acyl tails does a nice job of balancing the amount of space that the head group takes up, and that's why we find that kind of lipid in the lipid bilayer where it can pack evenly. If all three of the alcohols in the glycerol are occupied with fatty acyl tails, now the tails take up more space than the polar group, and it doesn't pack very well either, but it does just fine in a chaotic environment like a lipid droplet in a fat cell. So the precise thing we put on for the polar head group will provide the fine-tuning of how these lipids can then interact or form bilayers. So glycerophospholipids are one major group. Another important group are the sphingolipids. And sphingolipids are called that because they have a sphingosine backbone instead of a glycerol backbone. And this is sphingosine, if we look at it. And it's sort of a 3-carbon backbone-ish thing here to begin with a lipid tail hanging off here. It's not an acyl tail. You can see there's an alcohol there, and an alcohol sticking up there. So this would be sphingosine by itself. And then we can add onto that amine group an acyl group. So this would be an example of one sphingolipid. You've got the sphingosine backbone, like that. You've got the acyl group hanging off there. And then you have a phospho something or other up there. Here's a phosphate, in this case, choline. This happens to be named sphingomyelin because it's one of the significant lipids in myelin, the myelin sheath that packs around nerves and makes it possible for nerve conduction to be so efficient. So you have the same general features as a glycerophospholipid. You have effectively a 3-carbon backbone, you have 2 lipid tails, and you have a polar head group with phosphate and then a choice of what else. But the fact that it's coming off of a somewhat different chemistry in here and here allows the cell or the body to use it in appropriately different ways. They're not identical in what they will do for the cell. Another lipid that is very important to membranes and to other things, although it's not as abundant as the glycerophospholipids, is cholesterol. Cholesterol is on a completely different design than the fatty acids or the sphingomyelin or the sphingolipids. Cholesterol has a fused ring structure like this, 3 6-membered rings and 1 5-membered ring. It's got this tail hanging off of it that sort of is free to flap in the breeze and so on, but is branched, so that makes it a little more awkward. And it has this ittle bitty, itsy bitsy teeny weeny polar group. So where the other membrane lipids had phosphate and stuff, Cholesterol only has that little alcohol sticking off. And just as you might remember, tyrosine could sometimes hide its phenyl ring inside the protein and stick a toe of its alcohol group out at the surface. That's sort of the way cholesterol inserts into membranes. Most of it is buried in the lipid sea with just that little nose of its alcohol group sticking above the lipid sea. Now, if you can't remember it, it won't be my fault. The other thing that you do with cholesterol is it's a precursor for a number of other things. So there's families of steroids, not just one or two, but there's a whole family over here of the glucomineralocorticoids that are involved with regulating sugar and mineral metabolism. And then the other big family that you may be more familiar with of the sex hormones of testosterone and estrogen and all those guys. So it's a very busy molecule. Okay. All of the lipids that we've talked about so far are ones that are present in enough bulk to be used structurally to make up part of that lipid bilayer of a membrane to provide that platform for whatever else a membrane is going to do. But the body uses a number of other lipids in other interesting ways as well. There's a whole family of lipids called the isoprenoids because they're derived or are analogous to polymers of isoprene. So this is a 5-carbon, 1, 2, 3, 4, 5-carbon molecule with a couple of double bonds in there. And you can imagine that the chemistries available through that are not going to be the same as something that is simply unsaturated linear fatty acid or even one with a couple of double bonds in it. Here we have ubiquinone, and we're going to see ubiquinone again. It's a very important electron carrier. And its tail here is a string of 10 of these isoprenoid units. And if you kind of imagine what that probably looks like with its double bond being semi-rigid and its branched, its methyl group sticking off, it's definitely a little more kinky than a typical fatty acid would be. You can also, I think, see some of the analogy here in phylloquinone. This is vitamin K, and it's essential for producing clotting factors. That's what the K comes from. It was discovered by Germans who spell clotting with a K. It's phylloquinone because you get it from leafy green vegetables. But you see those branches, and so it's an odd one again. Retinol is another one in this family, and you probably recognize that as vitamin A. It plays a role in light transduction. So all of these things are things where you don't need a whole lot of them, but you do need enough to do the job. Vitamin D can also be generated in small amounts from a membrane lipid, in this case from cholesterol. And so if you look at this structure, you can see how it's fairly similar to cholesterol, but you've broken one of the rings right there. And that ring is sufficiently fragile that you can break it with sunlight. And so you see a gradient of skin darkness from the equator up to the poles, partly because we need a certain amount of vitamin D, but not a huge amount. And people who had the darkness of people growing at the equator who went north started to get vitamin D deficiency, so it's selected for having paler skin where you could make more vitamin D and get the right amount. In the Victorian period, women who were dressed completely head to toe, because you don't want to have a man shocked by seeing your wrists or your ankles, were almost never, ever exposed to sunlight. And they didn't know about supplementing with vitamin D. And during each pregnancy, these women would have to provide calcium from their own bodies for the pregnancy. So after 8 or 10 of the pregnancies, many of the women in the Victorian period started to have things like broken bones or 2 inches shorter than when they started having kids and so on, principally traceable to the fact that they couldn't make vitamin D and capture the calcium from their food. Before we talk about membrane fluidity, this might be a good place to take a small break, and I'm going to ask Susan to help me pass around some file cards. We often ask students to write exam questions or study questions for tests, but I think if we wait till the last minute, we're not going to have you remember enough about enzymes to be able to do that. So I'm going to pass around some file cards and invite you to write, if you want, some either study question or exam question, and if you want, you can tell me which, or you can just make them up. And if you don't want to write a question, you can just write your name on the back, and it'll still count. So, If there are cards left over, please make sure they get back to the front. Can I ask one of you to take some down to those poor souls over there? There you go. Great. 2, 3, 4. We will get you some cards. Do we have some extra cards somewhere? We need some up here. So what I'd like you to think about are study/exam questions about enzyme mechanisms or about enzyme kinetics, or perhaps a question that taps into both of those. And then we'll keep those in mind and use those as part of the review questions, and if they're good ones, on the test itself. Does anybody who wants a card have one? Okay. And if you don't want to write a question, that's fine. You can still put your name on it. You can talk to each other, by the way, if you want to cross-check with each other whether you think it's a reasonable question or not. While you're working on writing those down, I'll try not to distract you too much, but I want to emphasize something I said in the email I hope you received. There was a correction made on the Wednesday handout, which I hope you received. And the two nice students who corrected me came up after class again. And I try to make my handouts correct. I'm human. There will probably be errors despite that. My new promise is if you correct me in class rather than after class, I will give bonus points. I would really rather have it corrected sooner rather than later, so don't be shy. Mm-hmm. Mm-hmm. Does anybody need more time for writing your questions? Okay, couple more seconds there. Mm. Mm. Mm-hmm. All right. Feel free to go on writing, but I think we'll get back into the flow of the lecture if you don't mind too much. You're certainly welcome to stay a minute or two at the end if you have more you want to write. On your way out, just leave them on the table there, okay? Either with or without a question, but as long as we know you're here, and we'll keep that in mind. Let's talk a little bit about membrane fluidity. Part of what makes a membrane so good at what it does is the fact that it is not a fixed structure. It is a structure that's constantly changing its shape in subtle, interesting ways. So the lipids in the membrane, in the plane of the membrane, are constantly free to move around and sort of elbow each other around. And so the dynamic character of this lipid bilayer is a consequence of the lipids that are in it. So what kinds of lipids you use, that will determine how fluid a membrane is. This is a good example of the typical dimensions, and this diagram is fortunately honest enough to point out that it's not a rigid line and it's not just straight things in there. Some of the things that probably would make sense to you is that the longer the fatty acid chain, the more rigid it is, that the longer it is, the higher a melting point it has, and so the more rigid it'll tend to make it. An unsaturated fat will have a lower melting point. And one way you can keep all of this straight in your mind is think about butter and olive oil and canola oil. I'm assuming at least some of you cook. The butter is solid at room temperature. The olive oil and the canola oil are both liquid at room temperature. You put it's fairly solid at room temperature, depending on whether it's July in Iowa. You put the olive oil and the canola oil in the fridge, and the olive oil will become kind of a milky solid. It'll kind of look like olive oil butter, but the canola oil will still pour freely. And the differences among these are that butter is almost purely saturated. Olive oil is mono-unsaturated, it has a single unsaturated bond or double bond. And the canola oil has a lot of polyunsaturates. And so here you have a very direct example of the effect of saturation on melting point, and it has the exact same effect on the saturation of the membranes and the fluidity of the membranes. Now, in this little picture, we're trying to capture something about an issue that's very much making the news recently, which is trans fats. If you start off with polyunsaturated oils like corn oil or canola oil, and you want to make something that's a little bit solid, but not terrifically solid, like maybe margarine, the old way of doing this was to bubble hydrogen gas through the polyunsaturate until you got some of those things converted into single bonds and some of them not. But in the process, you could end up with some double bonds that were unnatural. So this is a trans double bond. That is the bond. If my torso is the double bond, the one bond is coming forward and the other is like that. So this is a trans double bond. Sorry, this is a cis double bond. They're both on the same side. In a trans fatty acid, they go back and forth like this. And this is a cis double bond, or the natural kind. It's kinky. And kinky in this case is good. Kinky is what makes things makes the lipid layer fluid. In a trans double bond like that, it looks like it's saturated. It may be a double bond, but it's going to be held out in a more rigid, straight structure. It will tend to raise the melting point. It also seems to be handled differently by the body. And we have a growing body of evidence that a trans fat is sort of like a super saturated fat in terms of its effect on the body. So this membrane fluidity thing is of more than just abstract interest or importance. Any questions about the cis-trans? Yes? Usually what happens is they're taking polyunsaturates to monounsaturates. And polyunsaturates, in polyunsaturates, those double bonds can break and reform, or can oxidize with each other and reform and polymerize. And when they do that, you end up with, rancid whatever it is. That ability for polyunsaturates to, oxidize and cross-link and everything is what makes linseed oil paint work. Is the polyunsaturated fats in the linseed oil are oxidizing and polymerizing. The trans fats are generally, by the time they're used for things like this, a monounsaturated, so they have less potential for doing that. And I think that also in that trans state, they may also be less susceptible to oxidation, even compared to a monounsaturated with a cis bond. They also have textural advantages. They used to say that, it makes the cookies have a better mouthfeel and things like that, but they're getting better at finding ways around that. But polyunsaturates will oxidize a lot more easily than monounsaturates. That's why you need to be nicer to your canola oil and your walnut oil than to your olive oil. Unless, of course, it's virgin olive oil. The fluidity of membranes means a couple of things. One thing it means is that the lipids themselves can diffuse. They can move by the random thermal energy in the environment. And that diffusion, if it's from one place in the plane of the membrane to somewhere else in the same plane of the membrane, is very rapid. This is called lateral diffusion, side-to-side diffusion. So that's very rapid. But as you can imagine, diffusion the other way, of going from one leaflet over to the other leaflet, which we call transverse diffusion or a flip-flop, is not very fast spontaneously, because that polar head group would have to dive through that lipid sea and come out the other side. So going from one side of the leaflet to the opposite leaflet does not happen very easily. And there are, in fact, enzymes that will do that, translocases that will cause it to translocase. They're also sometimes called flipases because they flip it from one leaflet to the other. So lateral diffusion, rapid; transverse diffusion, slow. In addition to these freely diffusing lipids that can simply move side to side in the plane of the membrane very easily, we're discovering that there are also patches in the membrane of lipids that like to travel together, that do not easily dissociate from each other, but travel as kind of a cluster. And these are called rafts, that they have a special lipid composition that is distinctive. They also have a special protein composition that's distinctive. And so it seems to be a way for cells to create a functionally specialized region within the lipid membrane. And these rafts tend to have some unusual components. So for instance, these are a couple of components that are enriched in these rafts. This is a cerebroside, and you can see here it has a sugar hanging off of its end. You have here a ganglioside with a complex sugar hanging off of its end. And then finally, overall, the composition of the membrane is tweaked and tuned with cholesterol, because cholesterol can act from both sides of it. So it can raise the melting point of things that are unsaturated and lower the melting point of things that are saturated. And if you think about it, so if you think about something that's got a lot of saturated fatty acids in it, the cholesterol sticks into that and makes it hard for them to pack, so it will lower the melting point. Whereas if you have a lot of unsaturated fatty acids, you can imagine the cholesterol's in there and it's this broad flat thing, and it tends to cut down the amount that they can bang around and raise the melting point. So cholesterol kind of tunes the membrane. You can think of it as kind of the Goldilocks component, not too fluid, not too tight, just right. So let's take a little look at these. Here's three lipids, a cis-oleate, a trans-oleate, and linoleate. Okay. So if you do the math on cis-oleate, you have 7, 7, 14, 15, 16, 17, 18. So this is an 18 carbon fatty acid. This is the trans-oleate, which is same number of carbons, but in this case the trans double bond. And then linoleate, which has 2 double bonds. Which of these would have the highest melting point? Which of these would have the lowest melting point? You can talk to each other if you want while you think about it. So anyone want to take a stab at which of these has the highest melting point? B? C? So you're going to suggest that C has the highest melting point. It takes the most heat to make it melt. Would you like to explain your answer? Okay, so it has 2 double bonds. And generally, what effect do double bonds have on the melting point? They tend to lower it, because the more double bonds, the more kinky it is, the harder it is to pack, the easier it is for it to... I'm sorry, I'm getting scrambled again. You're right, it has the highest. You know, this is the problem with some of biological things. You have the... No, it's the lowest melting point. It's the lowest melting point. Butter, for instance, is saturated. It has a high melting point. It'll stay solid at room temperature. Canola oil is polyunsaturated. It will be liquid even in the fridge. So it melts below 40 or whatever you keep your fridge at. So linoleate with its 2 double bonds, it's more kinky. It will stay liquid down to a lower temperature. Okay? So now we've got linoleate straightened out. Yeah. Are they what? Yeah, they would be cis unless otherwise specified. So in other words, the natural condition of double bonds in biological compounds, or at least fatty acids, is in the cis form, and they'll only be trans if they specify it. Good question. So do we want to try and sort out then between the other two, which one will have the highest melting point? Okay. We're hearing some calls for B. Would anybody like to justify B as having the highest melting point? Well, that's a nice way of putting it. Why would it have more surface area, though, than A?
Because A kind of like takes it.
Uh-huh. That's certainly a way of thinking about it. It's also going to have this ease of packing, because with the trans fatty acid, it'll be more straight. It will be capable of being straight, whereas a cis double bond, you can never make that puppy completely straight. It's always going to have a kink at the double bond. The rest of the stuff may be straight, but the kink won't. So, and I have one here about McDonald's, but you know, since I made the slide, McDonald's has tried to clean up, I think. So they're claiming they don't have trans fats anymore. We'll see. There's somebody who was saying that in the fryers that McDonald's has, they cannot change all of the oil at once. They have to leave about a quarter of it behind, because it's the oxidized lipids that make that wonderful taste you like so much. So if you change all the lipid at once, that's too fresh, it's not oxidized enough. So, food science, all kinds of amazing things. Okay. But lipids alone are not enough to make a membrane. We have to keep in mind that lipids alone are relatively rough instruments for getting something done. And so the other important component of biological membranes is the proteins. Most diagrams of membranes are like this one, where they show kind of a sprinkling of protein. But you should keep in mind that about half the mass of membranes is protein. And if you think about that in sort of a spatial way, think of a checkerboard with the black and the white squares. That's telling you how packed the proteins are in a typical membrane. And so these diagrams that are more typical to make it easier to see underestimate the density of the protein. Now, apart from the self-sealing character and the barrier character, most of the functions that you think of cells performing with their membranes are going to be due to the proteins. And we see membrane proteins of several kinds. We see some that go clear through the lipid bilayer, and these are called integral membrane proteins because they're integral, they're an intact, continuous part of the membrane. We see some other membrane proteins that kind of sit on the edge of it, interacting with the polar head groups there. And we see a third kind, relatively recently described, that have a little lipid tail. There's a lipid covalently attached to the protein, and that lipid tail can stick down into the lipid bilayer. To some extent, we can sort these guys out by what it takes to get them out. So for instance, the peripheral membrane proteins are just interacting through electrostatic interactions. And so high salt will often strip peripheral membrane proteins off of a membrane. Integral membrane proteins are clearly all the way through it, and to get them clean, you usually have to use strong detergents. One of the reasons why it's so hard to study them. Their new methods start to make it possible, but for many years it was impossible to do X-ray crystallography on them. And then these guys, with their lipid tail, are kind of betwixt and between. Some of them will come out with mild detergents. Some of them need a little stronger, but salt alone is usually not enough. Okay. Integral membrane proteins have to have some way of getting from one side to the other. And there are two broad strategies for doing this. One is to use a bunch of alpha helices. And so you have one or several alpha helices that go clear across from the hydrophilic side through the lipid part back out the other side. And so here is a skeleton backbone representation of one of these alpha helices. And you can imagine that when you have this bundle of alpha helices, that the side chains that are in contact with the lipid have to be hydrophobic. And so this is the big exception to the general rule that hydrophobic side chains are on the interior of a protein. Here, they have to be in contact with the lipid. And I'll defend the previous statement about it as a generality because lipid membrane proteins are not a very large fraction of all proteins. They're a few percent. And if you do the math on it, it takes about 20 residues to make a helix long enough to go from one side to the other. And so this gives us a way to recognize transmembrane spans with some accuracy, but not perfect accuracy. So if you look at this amino acid sequence here, this is an example of a membrane-spanning alpha helix. And if you look at it, it's really rather hydrophobic. I mean, about the least hydrophobic thing in here is alanine or glycine, but you've got leucine and tyrosine and threonine. Well, threonine is not so much, but a lot of hydrophobic stuff in there. And there are computer programs that can go through protein sequences and make guesses, educated guesses, but guesses, at where the membrane-spanning portions of that protein are. An alpha helix about 20 residues long. So each of these alpha helices would be about 20 residues long. Another strategy is to use what are called beta barrels. And we're going to see beta barrels in other contexts like enzymes, but they are certainly found in membrane proteins too. So in this case, you have beta pleated sheets that are side by side and a little twist to them until they make a complete cylinder of beta sheet. And so this is an example of a beta barrel where all the secondary structure you can see is beta pleated sheet. So the hydrogen bonds are going side by side like that and side by side like that. And the smallest beta barrel that has been observed is an 8-strand beta barrel. Some of them are very big with up to 20. Obviously, that'll make a difference to overall configuration. And if you think about it, with the beta sheet, the head groups, the side chains stick out on each side of the secondary structure. So it's only the residues that are sticking in contact with the lipid part of the lipid bilayer that need to be hydrophobic. The side chains that are sticking into the interior of the beta barrel can be hydrophilic. And we'll see what that does for us. A common feature is that you see here, if you sort of look at the decoration here, is seeing aromatic side chains near the edges of the barrel. Not a perfect thing, but they tend to be there. And so it's not as easy to pick out as the alpha helices for that reason. So which would need a longer stretch of residues to span the lipid bilayer, an alpha helix or a beta pleated sheet? Want to justify yourself? Oh, I think that's actually more extended. If you think about an alpha helix, you wind to get there, whereas to make a beta pleated sheet, you're almost as extended as that backbone can be. So it takes less than 20 residues of a beta pleated sheet to get from one side to the other. There are a rule of an angle, and that may be what's slightly misleading there, but the fact that beta pleated sheets are quite extended compared to alpha helices overcomes that. Now, the other thing we talked about were membrane proteins that have little tails sticking off of them, a lipid tail. And there's quite a variety of these tails. These tails are often attached to a sulfur-containing side chain at a cis, for instance. And you can see in these two particular examples the sulfur of the cysteine that's the anchor there. Some of them are just simple, unbranched lipids like, myristil or palmitoyl. These are just simple, straight hydrocarbons. Some of them are the branched and desaturated stuff of the isoprene family. So here you can see the side chains and the double bonds we saw in the isoprenoids. And there's another family, the glycosyl phosphoninositides, where there's a sugar group joining between the lipid part and the protein part. So there's a kind of a buffer between the whole thing. These turn out to be important because some of the cancer genes are mediated through proteins who are reversibly attached, in some cases, to the membrane through such tails. And if they're mutated so that they can't be attached to the membrane or they can't be detached from the membrane, that can either turn them on or turn them off on a constitutive basis and make them virulent. Sorry. So we're going to get back. This is a place where I differ somewhat with the book. Okay. I'm sure you've heard before this if you've had your biology class. Maybe it's out of your head by now. The phrase "the fluid mosaic model." The fluid mosaic model was a model for how membranes are made that Singer came up with in the 70s. And the general idea was that you have a lipid sea of a lipid bilayer, and you have proteins in it that then are free to move in this lipid sea. So it's a mosaic of proteins, but there's this sort of lipid sea for them to move in. And that was a reasonable place to begin, and there was some evidence for it in the system he began with, which is red blood cells. But it is really not a very representative model for membranes in general. The lipid part is fine. The membrane lipids, for the most part, except for the raft ones, are free to move side to side at the rate of diffusion or close to it. So it's a lipid mosaic it's a fluid mosaic as far as the lipids are concerned. The proteins, on the other hand, do not move at the rate of diffusion under most circumstances. Some of them don't move at all. They are fixed in place and do not rearrange. Some of them are what they call corralled, where they'll bump around in an area for a while and not go out of there. And some will move, but more slowly. They may go anywhere in the plane of the membrane, but get there at 100 or 1,000 times more slower than true diffusion would do. And interestingly, the only place where we see membrane proteins truly behaving as if they're diffusing is in little bubbles that come off of cells called blebs, where there's no cytoskeleton underneath the bleb. The bleb is essentially a pure lipid bubble, and in that little lipid bubble, there the proteins diffuse. But for virtually all of membranes in cells, it's really a mixture of a fluid mosaic as far as the lipids go, a solid or semi-solid mosaic as far as the proteins go. And I think this diagram will kind of highlight for you why this is true. This happens to be a picture taken from a red blood cell article. You don't need to memorize the names. But what you can see here is there are several different kinds of integral membrane proteins here. And these integral membrane proteins are interacting with elements of the cytoskeleton. And the proteins that help the membrane proteins interact with the cytoskeleton are complex and getting better known every year. And so if this is the structure of the membrane, it's not surprising that the membrane proteins are not free to diffuse throughout the thing. Again, this picture is good for showing you how the membrane proteins are anchored. The actual density of proteins would be close to 20 times greater than this. So, even for the proteins that can move some, we can show that they are constrained in an area, and this is a picture of that showing you how these are proteins where an individual protein molecule has been trapped for a period of time. And you can see how it's bouncing around in a little area, but it's not getting away, if you like. And so each of these patches represents a protein that's fixed in a little corral and not able to escape. And again, others, as I said, don't move at all. Interesting example of that, if you look at your muscles, everything you're familiar with doing physically that's moderately complicated, like riding a bike or skiing or whatever, is fixed into your muscles as a pattern of proteins. And those patterns will last for years. So clearly this process of anchoring things in the membranes is efficient and durable. Oh, it's 4:20. Listen, have a great weekend. Don't forget to leave your card down here, okay? I'll leave it there.